Hi, and welcome to Kent County High School Science, AP Biology, free response question number 4. This is going to be a 10-point question, so please expect to read a little bit more, expect to analyze a little bit more, and certainly you're going to write a little bit more than for a 3 or 4-point question. Also, this question is going to feature some content you've never seen before. So if you're saying to yourself, well, how can I answer a question if I've never seen the content? You're always going to see content on an AP exam that you've not learned in class. The focus is on what are called science practices, reading graphs, doing a little bit of math, analyzing data, posing a question, generating a testable hypothesis, do the results justify the conclusion that was reached. So everyone can do the science practices. So we're doing this for practice. Let's go. So here's the graph that goes along with the question. Take a moment. What's on the x-axis? Time in hours. What's on the y-axis? Seed germination. Germination is when a seed begins to sprout, a root emerges, and then a shoot. So look, we have percentage there, we have hours and time on the x-axis. The caption, the effect of characins, K-A-R, and trimethylbutyloids, TMB, on seed germination and lactuca plants. Error bars represent plus or minus 2 standard errors of the mean. You don't need to know what a lactuca plant is. You just need to know it's a plant. It produces seeds. You don't need to know what characins and TMBs are. That's going to be explained to you. So the stem of this information reads as follows. Fires frequently occur in some ecosystems and can destroy all above ground vegetation. Many species of plants in these ecosystems respond to compounds in smoke that regulate seed germination after a major fire. Characins, K-A-R, and trimethylbutyloids, TMB, are water-soluble compounds found in smoke that are deposited in the soil as a result of a fire. KAR and TMB bind to receptor proteins in a seed. In a study on the effects of smoke on seeds, researchers recorded the timing and percent of seed germination in the presence of various combinations of KAR and TMB. The results are shown in Figure 1. Now the question continues. There's a data table. In a second investigation into the effect of available water on seed germination after a fire, researchers treated seeds with KAR or TMB. The treated seeds were then divided into 2 treatment groups. One group received a water rinse, and the other group received no water rinse. The seeds were then incubated along with a group of control seeds that were not treated. Remember, incubated just means they sat for a period of time. The results are shown in the table. So take a moment, pause the video, study the table. Look at each column. We have groups 1 through 6. Note which ones are the control. Note the positive and negative signs to see what treatment was applied or not. Notice the water column. Was a water rinse done or not? And take a moment and look at the germination result that came about because of these different conditions. Now, here's your first question. You're going to write the word A, and then you're going to answer. The researchers made the following claims about the effect of KAR and the effect of TMB on seed germination relative to the control treatment. We have 4 bullet points. KAR alone affects timing of seed germination. KAR alone affects the percentage of seeds that germinate. TMB alone affects the timing of seed germination. TMB alone affects the percentage of seeds that germinate. Provide support using data from Figure 1 for each of the researcher's claims. Please type A, and then you need to write a minimum of 4 complete sentences to answer this question. More is okay, but you're going to need at least 4 because you need to address each bullet point. Go. Okay, part B. I gave you the data table and the caption that went along with it. Part B asks the AP student to make a claim about the effect of rinsing on the binding of KAR to the receptor on the seed and on the effect of rinsing on the binding of TMB to the seed. Identify the appropriate treatment group and results from the data table that, when compared with the controls, provide support for each claim. It's going to take you several sentences to do that. Pause the video, go. Finally, there's a part C. I gave you both the graph and the data table. Part C, there is intense competition by plants to successfully colonize areas that have been recently cleared by a fire. Describe one advantage of KAR regulation and one advantage of TMB regulation to plants that live in an ecosystem with regular fires. Go. Now let's score. So here's the rubric. For part A, there are your 4 bullet points. You could earn 1 point for saying KAR affects timing. The germination started sooner, quicker, faster, earlier. That's a point. KAR affects the percentage of seeds that germinate. Well, if you look at the graph and look at the KAR alone, that's the highest percentage of germination of any condition. TMB affects timing. You need to say as part of your answer, the germination started later or slower. Look down all the way at the very bottom at the TMB only line. And TMB affects percentage. Well, it looks to me like the TMB treated plants were somewhere a little bit between 10 and 20%. That was the lowest percent of seeds that germinated. For part B, you were making a claim about the rinsing on the binding of KAR to the receptor of the seed and the effect of rinsing on binding of TMB on the receptor in the seed. You need to identify a control group and you need to identify the proper experimental group. So that's going to provide you with support. So if you claim that KAR remains bound after rinsing, or you said rinsing doesn't affect the KAR binding, you need to compare group 2 to group 5 with no rins and with a rins, and notice that that is different than control groups 1 and 4. If you said that for the other part of it, TMB does not remain bound or the rinsing affected the TMB binding, you're comparing group 3 and group 6 with groups 1 and group 4. But notice with group 6 you had the same outcome as you did with group 4. So you're looking at with the water and without the water as compared to control groups. And finally, there's intense competition by plants to colonize. What's an advantage of KAR regulation and what's an advantage of TMB regulation? For advantage of KAR regulation, you could say germination occurs at times of increased resources. Plants with KAR regulation can outcompete other plants, or germination occurs when fewer competitors are present because these plants are germinating very early. The advantage of the TMB regulation, you might have had a harder time thinking of this, but those plants can stay dormant and not germinate until enough water is available. Or you could say they won't germinate in a dry environment, which means maybe the plant won't necessarily survive long enough to reproduce. Okay? So, let's take a look at a student sample. Part A. When only KAR was present, the seeds germinated at 12 hours, which is almost half the time for the control seeds. When only KAR was present, 100% of the seeds germinated. When only 80% of the control seeds germinated. When only TMB was present, the seeds germinated 8 hours after the control seeds at 24 instead of 21. And when only TMB was present, only 20% of the seeds germinated versus 80% in the control. They're getting some points there. Seeds that had KAR present and were RINs began germinating very quickly, faster than the control with water presence. Water presence sends a signal which removes the inhibitor and allows the KAR to bind to the receptor and start germinating. We don't necessarily know that, but I suppose that's possible. Seeds that had TMB present and were RINs began germinating at the same time as the control. The water present also sends a signal for germination to begin and for the inhibitor to stop binding to the active site. They're reading a little bit more into this than really is. They have earned 4 points for part A, but it's all that material up at the top that is simply saying KAR seeds germinated sooner, higher percentage of germination, and TMB germinated later and a lower percentage. An advantage for KAR regulation, seeds germinate faster, more of them germinate. In the race to take over space, this is an advantage as they germinate faster, grow faster than other seeds without KAR regulation. The TMB prevents the seeds from growing when there's enough water, nutrients present for the plant to survive. This student ended up earning 6 points. You'll notice that they really didn't get any points for part B because they were not comparing to control groups and identifying the groups that they needed to mention as part of the rubric. So still 6 points, not bad. Let's take a look at this one. KAR plants, KAR alone affects the timing of seed germination. According to the graph, KAR seeds germinate about 3 hours earlier. There's a point. KAR alone affects the percentage of seeds that germinate, shows almost a 20% increase when compared with control. There's a point. TMB alone affects the timing of seed germination. According to the graph, TMB seeds germinated almost 6 hours later. Point. TMB alone affects the percentage of seeds that germinate. Graph shows almost 65% decrease. There's a point. When rinsing the experimental seeds in water, TMB rins off while KAR didn't. In the first test, both KAR and look, they're using numbers of which groups that they're comparing. They differ from the control. In the second test, however, TMB results the same as the control and the KAR. Once again, they're identifying the number of the group they were talking about. TMB doesn't bind to the receptor proteins when RINs. KAR still binds to the receptor proteins even after being RINs. KAR regulation allows the seeds to grow more quickly, giving them an advantage when trying to spread and reproduce. TMB regulation conserves resources and energy for the seed, giving it a better chance of survival in harsh conditions such as after a fire. Once again, this was a pretty good job. This was an 8-point response, 8 out of 10 on a free response question. You ought to be absolutely jumping for joy. So look at what the average score was that year, 4.04 points out of 10. Fairly challenging question. So please reflect on how you did and what you learned this time about writing free response questions. Hope this was helpful.